Hi, I'm Alex Marras. I'm a postdoc at University of Chicago, and this covers my progress programming biomaterials, specifically DNA and polymers, to self-assemble into nanoscale engineering devices for molecular robotics and gene delivery. So first I want to take a look inside of our cells at nature's machinery. We see things like molecular motors, organelles, and lots of instances of sensing, actuation, and programmed motion. But if we think about life at the macro level, we have robotics that uses a lot of these same features, but it's made from different materials. And so ultimately, what I'd like to do is borrow some of these concepts from macro scale mechanical engineering and from chemical engineering to create something with a level of complexity that we see at the molecular level in nature. And this is the goal that I've been working towards and plan to continue towards throughout my independent career. So I'm going to do this using some of the same building blocks that nature does. One of them being DNA. DNA is highly predictable, it's easily synthesized, and it can be used for structural assemblies like I'm showing here. So Ned Seaman pioneered this field and showed that through sequence design, you can create these structural elements that can bridge helices together. And this was this was expanded upon with DNA origami, showing this incredible geometric precision down to the sub nanometer level. And another aspect of DNA nanotechnology that I think sometimes is overlooked is actually the mechanical design space. So single-stranded DNA is very flexible, but once you hybridize it to form double-stranded DNA, you're increasing the stiffness drastically. And then through sequence design, you can actually bundle these and make these double-stranded helix bundles that can traverse orders of magnitude of persistence length. And these can actually be dynamic too through hybridization, going back and forth between something that's stiff or rigid. This combined with the geometric precision that I just talked about makes DNA a very attractive material. It's unlike any other soft material in terms of the design space. So we use these concepts first to create a nanoscale hinge. So this is a basic type of rotational motion. We have these 18 helix bundles that are very stiff and connected together with single-stranded DNA forming a hinge axis. And we can actually change the rotational stiffness of this by changing these connections on the back, giving us different programmable hinges. And we made something for linear motion. We assembled these two rigid components concentrically connected by single-stranded DNA to give us linear motion, first exhibiting rotational and then linear motion. And then following this, showing more complex motion like 2D motion and elastic components, and translating this up even into multi-joint mechanisms like this crank slider that couples rotational and linear motion, or devices that have many different possible conformations. All to show for the first time that you can really use DNA as a material for molecular robotics and have this vast mechanical and geometrical control for something like this. But we wanted to, but we also wanted to be able to control the motion or control the configuration of these devices. So I modified the design of this 4-bar linkage to have these pieces of single-stranded DNA hanging off of the rigid arms, acting as targets for another piece of DNA, like these green pieces that combined to opposite arms and then pull this together. And then you could add another set of DNA strands that can release this, changing it back into the open configuration. We used a bulk fluorescence quenching assay to show that this actuation happens in a minute or a couple minutes time scale. And then more recently, we created, or we developed another type of actuation using the same sort of design where we have these single-stranded handles hanging off of the rigid arms, only now these are much shorter and actually are not gonna, and these are gonna bind to each other now, but they're not strong enough under low salt conditions to actually overcome that electrostatic repulsion from the arms and close this hinge. But what we can do is change the solution conditions to add more salt to buffer these or screen these repulsion interactions and effectively strengthen this cooperative binding between all of these shorter, weaker connections to close the hinge. We can tune the actuation response by changing the pattern or the length and effectively the strength of these connections to explore and tune the actuation response to different ranges of magnesium concentration shown here and actually other cations as well. And one big advantage of this technique is that you're not relying on extra DNA to hybridize or displace, and instead it's reacting to buffer conditions, which happens much, much faster, as I'm showing here with this single molecule FRET assay. As I change the buffer conditions over a few seconds, these transitions from open to closed happen on millisecond timescales. So this is much faster and just a very different way of controlling the motion of these devices. So far I've shown this framework we've established for molecular robotics, self-assembled nanomachines, and ways to control their motion. But I also want to talk about DNA and nucleic acids in general as a genetic material, more of a traditional sense. And specifically, we're interested in oligonucleotides, or short pieces of nucleic acids. And these are interesting for things like gene therapy or silencing, and have immense potential, and people have been working towards this for a long time, but it's really limited by delivery right now. Basically, we need to protect this cargo and we need to control the transport and get it to where we want it to go. And so these are kind of the two gold standards right now, one being chemically modifying the backbone of the nucleic acid to prevent nuclease degradation, or encapsulating these into different types of hydrophobically driven nanoparticle assemblies. Both of these have been successful in certain circumstances, but they also have their limitations. And so the method that we're going to use is polyelectrolytes. These are charge-driven assemblies of hydrophilic materials that allow for different cargos, distribution, and even targeting. And these have actually shown to be effective in cell studies, both in our lab and in some other labs. But we don't fully understand the assembly process yet, and we don't have a way to control the physical parameters that are ultimately going to be important for delivery and for distribution. So in my postdoc, I've set out to understand how the different physical and chemical parameters of each component of these assemblies affect the overall assembly and the stability of those assemblies. So an overview of a couple different studies here. The different things that we see is that one, we can control the morphology. We see with using single-stranded DNA, which again is very flexible, we get all these spherical nanoparticles, but once we use a double-stranded piece of DNA, these are actually stacking and creating these long worm-like cylinders across the board with different polymers and different lengths and all these different parameters. We're seeing this drastic difference due to the hybridization of the nucleic acids. In addition to this, we also see morphological changes due to the molecular structure of the nucleic acids. So when we look at DNA versus RNA, we see still spherical micelles in the single-stranded case. But then once we add this methyl group for this 2-methoxy modification, which is commonly used, it completely disrupts complexation. And now instead of nanoparticles, we see this complicated web of unimer complexes and really no nanoscale assemblies. However, once you add the phosphorothioate modification, another commonly used modification, this actually, in this case, will enable complexation again, and you do get micelles, albeit not the spherical micelles that we see in the other cases. So all this to say is that these molecular details here have a very important role in complexation and in these nanoparticle formations and assemblies. Another thing that we found is that the size of the particles is controlled largely by the length of the charge block of the block copolymer and is largely independent of the length of the nucleic acid. This is, and also there's some dependence, maybe a loose dependence on the peg size. This is actually another ongoing project, is to really determine exactly how these things are scaling due to the length of each parameter, which will enhance the design process in the future. But the big thing that we've seen so far is that the size of this nucleic acid does not affect the size of the micelle, but it's largely driven by the cationic block. And then the last thing that we see is the stability. We looked at two different cations. We looked at polylysine and PBBTMA, which is a synthetic polycation. And we actually see that polylysine is forming more stable complexes with DNA versus PBBTMA. We think this is likely due to sterics with the larger cation here. And for the nucleic acids, we see a drastic increase in complex stability due to the phosphorothioate modification compared to the natural backbone across the board here, which was actually pretty surprising for such a small change. But this is really important to understand, because if we're talking about delivery, we need to make a stable complex to get this to where we want it to go, but ultimately you want to be able to release it too. So we really need to understand how the different parameters that we're putting into these nanoparticles are going to control the stability and ultimately the release of the cargo. So I've highlighted a few ways that we can program biomaterial self-assembly for various engineering devices, from molecular robotics to polymeric delivery devices. And I think that this cumulative design space is really enormous. And so looking forward, I plan to take advantage of this geometric and mechanical precision of DNA and the scalability and robustness of polymer assemblies to overcome some of the limitations from each material and really turn these into viable construction materials. And I think that these can be used for sensing and delivery and multi-scale controllable materials. I'm happy to talk about this more offline. This is kind of an overview of where I see this heading. But yeah, feel free to reach out. With that, I want to thank the Castro Lab at Ohio State, as well as the Terrell Lab at University of Chicago, and the funding sources and contributors and collaborators to this work. And thank you very much for listening.